our next couple of conversations, I'm excited to talk about some of the periodic table of food initiatives foundational resources. I'm going to move to the podium where I'm supposed to go. And first, we're going to hear about the PTFI tools being launched from members of the team at Verso Biosciences, which, as you heard, provides technology solutions that streamline data integration, simplify analysis and interpretation, and accelerate discovery. So I'd love to invite Tracy Shafazada, I think I got it right almost, I'm sorry, the Director of Programs and Partnerships for Verso Biosciences to present. Round of applause, sorry. I know, I'm so sorry.
I have a hard time with it,
too. Do it again.
It's really tough. Hi, I'm Tracy Shafazada. Great to see everyone. This is so fun. What a wonderful morning so far. Okay. So we are here to share with you what are these tools that we keep talking about at the PTFI. And what I want to do first is I don't have to convince this group that the food we eat and the nutrients that it provides us are inextricably linked to our health. You guys are here because you already know that. What's interesting is we're also starting to see that the way a food is grown and collected and transported and stored and processed and even prepared actually does influence the planetary health as well. That's really interesting and something that I'm not sure many of us think about. Now we do, but maybe previously we hadn't. And when we think about what is in food, most of us are used to looking at a nutrition label, which you'll see on the left-hand side of the screen when I move the slide. Let's see. A nutrition label does tell a story about a food. It tells about the essential nutrients, the macronutrients, the micronutrients, and it's based on decades of research on establishing the minimum requirements and the upper limits of the nutrients that we know are in foods and that are required for us to grow and develop and survive. But that is not the full story of a food. If we think about the nutrition label as being maybe the table of contents of a story of food, then we can dive deep into each chapter of that table of contents to a deep, rich, beautiful story that's complicated, interesting, and actually quite fascinating. And that's what we are striving to do at the Periodic Table of Food Initiative. We are trying to tell the full story of a food. So not only the depth of the chapters of each of the sections of what you would find on a nutrition label, but also all of the external elements of that story. Who are the other characters? So things like the cultivar, the collection location, the growing conditions, like I said, the processing and the preparation, these all influence what is actually in a food. So an apple grown in one location actually has a very different composition, potentially, than an apple grown in another location because of these external factors. So we can answer some important questions based on the information on a nutrition label. And in fact, there's a lot of work that's been done to make a nutrition label more readable and understandable by a general audience. A lot of work has been done to improve these. But what questions could we ask of a food if we knew its full story? So I'm going to give you an example. So if we ask the question, what's in a plum? We can understand the total calories, we can understand the total fat, we can understand total protein, total carbohydrates, maybe even a little more detail, maybe amount of fiber, maybe amount of sugar, which is something that's of great interest these days on a nutrition label. And that is interesting information. But what we're trying to do is say, let's dive deep into the chapters of that table of contents and say, if you look at, let's say, carbohydrates, there are different aspects of carbohydrates that give us a tremendous amount of information about how that carbohydrate will be used in our body. There's fibers, there's sugars, there's starches, but even deeper than that, there's different structures and functions and linkages that we can understand about aspects of these carbohydrates and these fibers and these sugars. Each of them do something different in our body. Some of them promote health, some of them are deleterious to our health. We don't know that information until we capture it, and then we can study it, and then we can look at what that means in terms of long-term health implications. So what I really want us to do is think about the food that we eat and the food that we grow deserves us to understand its full story. And then the very first thing we have to do, the very first step, is to collect this information in a standardized and robust way, put it in one central location that everyone can have access to, and really make it the most complete and detailed and robust story of every food we can get our hands on the planet. And that's exactly what the PTFI is trying to do. We're aiming to do it, and today we get to talk about the first 500 foods and the deep stories within each of those. So my colleagues today, Steve Watkins and Qiming Qian, will be talking to you about the tools we've built to analyze each of these foods, the approach we've taken to standardize and distribute these tools so that this isn't a single location that can do all of this work. It's distributed across the world to labs that are already becoming partners of the PTFI. And then eventually, and probably the most important part, are the visualization tools that allow people to explore the data, compare foods to each other, compare different growing conditions among a particular kind of food, and really bring the stories to life. So with that, I will end and pass it on to my colleagues.
Thank you, Tracy. You
Sure.
can join over here.
Thank you.
Tracy. Hi, I'm Steve Watkins. The only thing I decided I really want from all of you today is to say that, yes, this group is nerdier than the ethnobotanists. So actually, you know, I'm really thrilled and grateful to be here. It was about 4 years ago that Roy was standing probably 15 feet right below where I'm standing, and he was laying out this, what I thought was this crazy vision, and Greg and Bruce and others and I were here. But over the course of a day, listening to Roy and others talk and a dinner that night, I became pretty convinced and that I wanted to be part of this, that this was an incredible vision and that we could probably get this done. So thank you for inviting us to be part of this. And really, right from the beginning, we had a mission, which was to build the world's greatest and deepest database of food composition. But to do that with some tactics and strategies that I thought were really interesting. One was to do it by democratizing the ability to generate this data, right? So those of you who aren't in the analytical chemistry field may not know that the way a lot of this works is for labs to generate these data, and it's very specific to the lab. It's this kind of cottage industry where people are producing their own results, and they aren't comparable and interoperable. So we wanted to build the tools that would let anybody generate data and do it in a way that we could bring all of that data back into a central resource that would grow with time, to let people compare their current data to the reference data, build this deep resource, and really bring, I think, analytical chemistry and natural product chemistry into the same sort of status as genomics and sequencing that have really powered a lot of what we've done in biology over the last decade. The challenge is natural product chemistry is a lot more complicated than sequencing, right? At least the chemistry of it, actually doing the work. The foods in particular have an incredible array of biomolecules in them. You've got lipids, and you've got sugars, and you've got proteins, small molecules. And all of these tools need their own systems for analyzing them. So we spent a lot of time developing tools that are focused on each of these disciplines. And we've kind of structured them, as you'll see, around the standard sort of food components: lipids, proteins, small molecules, minerals. So you'll see as we go. So we have tools now that are now being run in our centers of excellence around the world for lipid profiling, both at a traditional fatty acid composition level, as well as a very deep, what we call a lipidomics panel, which is measuring more than 3,000 individual lipid species. We have panels for carbohydrates based on some technology that some of our early partners at UC Davis developed, where we're looking at starches and sugars, but as Tracy mentioned, also the fibers, these things that are fueling our microbiome, very important nutrients. We haven't had the tools to break them down and look at the components and look at the chemical diversity and structures there before. We're doing that. We have an incredible small molecule program that I'll talk just a little bit about, but I know others today will speak more in a more detailed way about. And we have protein and mineral technology, which I know others will speak about as well. So we've developed the technology across the board. And how did we think about standardization in each of these areas? It wasn't that we needed to develop new technology per se. It was that we needed to standardize technology that was already working, right? And so we looked around the globe. We did a lot of landscaping for protocols, methods that people were already using that had proven effective for foods. And then we focused our efforts on designing and manufacturing, standardizing reagents, and building data pipelines, and doing the kinds of things that are necessary to distribute to people doing the work that allow us to standardize and harmonize the data that come back. So that's really where the PTFI's focus has been. We've manufactured reagent mixtures in each of these disciplines that we send to the labs that do the work. Qiming will tell you a little bit about the data pipelines and the kinds of tools we've put together that let labs just bring their data to us, and we process it and build the annotations. And then we spend a lot of time using these standardized tools to build what we call libraries, which are, can we purchase chemicals and run them through our system so that we know what they look like. We can repeatedly tell labs that this is what this molecule is over time. And all of this, again, Qiming will walk you through a little bit of this. We've tried to structure the results in a way that the community can understand them. So when we think of omics platforms, I think probably when most people think of genomics, it's tons of data, very complicated. You don't think of being able to dig into a database and really make much sense of it. But with foods, we know they're structured around some basic components: protein, lipid, carbohydrate, water, small molecules. And so you can see here, we've even in the branding, I think you can see with the PTFI, we've really structured the way we're organizing the data for people to interact with it around these disciplines where people, if you're just interested in how much lipid is in a food, you can look at the first level of these sorts of rings. But if you want to dig in and really go deep, you can click through and start to understand deeper and deeper level what's happening with the food. So we've thought quite a bit about how to structure it, but we know that there are many of you come from many different disciplines and have different ways that you would like to see data and structure it and understand it. So we're all ears in terms of how we think about that going forward. Okay, so this is my most nerdy part, but I know that there are bigger nerds to come today because I know this group. So one of the things that Roy, was big on from the beginning that I actually have to profess a little bit of skepticism about, I think, maybe, but that worked out beautifully, is this idea of dark matter of food. So foods are incredibly rich, full of chemicals, and most of which we have no idea what they are, right? Which is kind of amazing. So one of the things we realized early in this process is that because we had standardized these analytical tools, we could, in essence, sort of build locations. We could see chemicals that we knew were there with high confidence, even if we didn't know what they were, right? So the way we do that is that we had built these standardizing tools that allowed us to see a location on our platform where we're seeing the same result in multiple labs from the same food. So we're getting this consensus signal from our network. Hey, broccoli has these 4,000 compounds, and they're all the same in all of our labs, even though we don't know what they are. So once we had built that standardization, we realized that we could add a lot of layers of information about those molecules. We could run them through platforms where we just determine the formula of the molecule, its retention time on the platform. We could also fragment them with a technique called MSMS, where we could look at the structural information. We could collect information on their shape using a technology called IMS, inclusive cross-section data. And we could look to see where those pieces of information were shared in food types. Is it all brassica? Is it all in Apiaceae? Is it just in tomatoes, not in peppers? So we could assemble all this into a structured database, and that's what we've done. We have built a very large database of all of these chemicals, many of which are unknown currently to science, that allows us to start to structure this data. This has been called the dark matter. And the amazing thing is that we built this library with these initial reference foods, and it has more than 18,500 chemical entities in it. So just in our first pass with this first reference data set, we are detecting 18,500 small molecules in foods that many of which we don't know what they are. And so this is just a little bit of a teaser. I know Jessica and others will present more, but this is kind of a way to look at the data. Those of you familiar with genetics might recognize it as a Manhattan plot, where the y-axis is the abundance of the molecule, and then all 500 foods are sort of ordered on the x-axis, and they're ordered by botanical family or food family, and colored by the same. This is a compound we all, actually I think it's one of Selena's favorite compounds, quercetin. And so you can look at our database and actually look at the distribution of quercetin in these foods. Obviously it's in berries, it's in apples. We found it in allspice and sumac and some other unusual things we did. But I think one of the really exciting aspects of PTFI is that we have 18,500 of these sorts of plots. And I think Jessica and I have looked through it, and we spent hours like down the rat hole of like looking through plot after plot. It's kind of amazing. I mean, and you just find endless number of things. We found a resorcinol in only one of our beef samples, and I went back and it was the beef fed, or it was the grain fed, not the grass fed beef. And resorcinol is a grain phytosterol, polyphenol that was getting into meat. So there's just endless stories. But I think this is kind of a neat one. This is of 18,500 chemicals. This is one that in our 500 foods were only in brown lentils, not red lentils, not other types of legumes. Like this, you can find thousands of these stories where we are just beginning to understand the complexity of our food and understand its potential to drive applications. So we hope you get lost in the data as much as we did, and can help us begin to organize it and make sense of this, so that we can really bring this to action, as Raj says, make it useful. All right, thanks.
Thank you, Steve. You can join us over here if you'd like. We're now going to see what is a very special demo from Qiming Qian, the founder of Verso Biosciences. So over to you. I'm so excited about this.
Thanks, Danny. Co-founder of Verso Biosciences,
Sorry.
along with Steve and Troy. I'm super excited to share, as Selena called it, our baby. It's a baby that a lot of you in this room have been involved in birthing. And as Selena also said, be kind. All right. So as Tracy and Steve talked about, we are really expanding the frontiers of food composition analysis by measuring more of the components in our food in a more standardized way, right? So what I'm going to take you through is one interface for exploring the database of first 500 foods. Yesterday, I don't know if some people that were keeping an eye on me probably saw me on my phone a lot. And I was not posting TikToks of Hyla the Earth, wasn't on Instagram, but what I was doing was I was searching the database to see, oh, that food that was mentioned, do we have that in the database, right? So we have all these wheat samples in the database. Do we have the Emmer wheat that Dan Saladino mentioned, right? And then later on the food walk, we did a plant walk. Alex was taking us through, pulling things out of the ground and folding it up and tasting it. And so it was kind of searching for different food samples. Do we have that in the database? And so, there's a lot of samples that we have in the database, 500. There's an inspirational list of 1,600 foods that we're looking to profile. I had a very interesting conversation with my daughter last night. So she's a biological sciences major. And apparently for a final or a project, she has to memorize a bunch of genera, genuses for different species for this, I guess that's in the diet of this one bird, right? So I'm a software engineer by training, but we got a chance to connect on scientific names of foods, right? So she said, Zea mays. I'm like, I know that one. It's corn, right? And then I said, well, do you know Triticum estivum? Thank you. Wheat, right? So in the database, we can search by different scientific families. We can search by the scientific name of the food. And so we're going to go ahead and search for a quinoa sample here. So you see that we have a number of different quinoa samples. There's ones that are produced conventionally. There are ones that are produced organically. and some are dried and some are cooked. So we're going to go ahead and actually pick this one sample here. It was a field collection that was collected in the San Luis Valley region of Colorado here in the United States. And we'll take a look at what that food looks like. So one of the things I'll point out, our colleagues at CIAT Steve and Tracy both talked about how metadata is so important, right? We need to be able to compare these foods to each other. So we've curated all of these foods to the Food on Ontology. What that means is that when we take a look at a quinoa sample, we actually can compare it to samples that the global research community collects and make sure that we're actually talking about the same thing, which is harder than you might imagine, right? In addition, if we have information about the cultivar, we collect that, the method of the production, where it was collected, et cetera. Now, in the center plot here, you see kind of a basic picture of the composition of this food. On our nutrition labels, as Tracy spoke about, what we've got is kind of the macronutrients, right? It's essentially in this picture, it's kind of the first ring and the second ring. So we've got macronutrients like carbohydrates, protein, lipid, ash. But we also have, the second ring in that is what kinds of lipids. So the saturated fatty acids, monosaturated fatty acids, omega-6 fatty acids. Now, what the PTFI is doing is taking this like a lot deeper, right? So for example, for the lipids, we can drill in and see the specific monounsaturated fatty acids, right? And the specific omega-3 fatty acids that are part of this sample. Within the ash, we can take a look at the specific minerals, and we're profiling, I think, 26, Jackie, 26 different minerals and elements, including some heavy toxic chemicals, right? And then proteins, we're doing amino acid composition analysis. There's also a full proteomics analysis, so there's about 6,000 proteins in this particular quinoa sample, and we have information on those 6,000 proteins. It's apparent that this quinoa sample is mostly, or significantly carbohydrate, right? The question is, what does that really mean on a very, very deep level? So we see that the carbohydrates, we've got starch, sugar, and cellulose, and we could take a look at the non-cellulosic fibers that are within this carbohydrate. We can see what the sugars are that compose those fibers, and also, we can see what are the linkages, like how are those sugars actually linked together. And this, several folks have kind of alluded to this. It's very important how these linkages occur because particular bacteria within our gut microbiome actually feed on sugars that are linked in very particular ways, right? So having this information, which has previously not been cataloged, actually points to things like precision medicine or food as medicine, right? Where we can address particular deficiencies, particular needs in the human diet. All right, and of course we have the quantitative information as well. I want to kind of point out the metadata. So for collecting a bunch of standardized metadata, that's been kind of the work of a number of folks in CIAT to help us to get to that standardized place. And so, for example, for this particular collection, we know the latitude and longitude, the date of the collection, and that allows us to connect to external databases like weather databases, precipitation databases, and so it allows us to begin to build the evidence base for how climate change then affects food composition, right? And so that's really important. The other thing that Selena mentioned this morning and John mentioned is that these metadata modules are built out to actually be both modular and expandable. So there's a regenerative agriculture metadata module that's in the works. It was Michael Kotutwa Johnson, as well as Curtis, yesterday were talking about traditional knowledge. And so being able to attach traditional knowledge to these samples also so that we know that if a particular plant was used for particular purposes, we can connect that to the molecules in that plant and potentially make some of those connections for why that plant is used for those purposes. Or there's other plants that have that same molecule that could be used for similar purposes in a culturally relevant way. One of the questions that you might ask is how a particular sample might compare to other foods within the database. And so for that, I'm actually going to take a look at a hemp seed sample. We're going to pick this, take a look at, so we got a couple of hemp seeds, one that was, both of them were produced or purchased in a supermarket in Fort Collins, Colorado, which I assume is very close to where the Colorado State folks are from. And one was produced conventionally, one was produced organically. And so we'll take a look at this one here. So again, same basic picture. But we might want to ask, like, what's interesting or what's particularly interesting about this hemp seed? So the Z-score report is actually a way to help us visualize this. So we'll take a look at the Z-score report here. I'm going to adjust this a little bit so that we can kind of see a little bit better. So it turns out that there's this one molecule at the top, FA183N6. It's a sounds like Star Wars. It's an essential, it's an essential omega-6 fatty acid. And it is significantly enriched in hemp compared to everything else. So if we take a look at this, this is, for those biostatistic nerds among us, it's 15 standard deviations above the average, right? It just basically shows up in hemp. Fortunately, I spoke to Steve, and we, this is a particular fatty acid that is only produced, or is produced in very few plants. Hemp is one of the few plants in which it can be produced. Okay, I want to take just one moment to talk a little bit about the dark matter of food, exploring the dark matter of food. So the metabolomics method is the PTFI method for exploring small molecules within food. So it takes a very broad spectrum look at all the molecules in food. And as Steve spoke about, we've identified 18,500 different molecules, and we're looking for those molecules in every single one of our food samples, right? So if I take a look at a particular food sample here, go back to our quinoa sample, and this is what quinoa kind of looks like. I'll bump it up a little bit for y'all. So here we see an essential omega-3 fatty acid, alpha-linolenic acid, which is known to be in quinoa. We also see crustic disone. So this is something that a software engineer doesn't expect to find out about. But crustic disone is a molting hormone that's found in crustaceans. And that's the name crustic disone. It's also found in quinoa, and the literature shows that. And what happens is that insects consume these plants, and then it induces them to molt. But what's interesting is because we're searching for those 18,500 molecules across all of our food samples, we also see that there's dozens and dozens of molecules that are much higher intensity in quinoa than these known molecules, right? So let's take a look at one of these. We'll take a look at kind of the top hit here and do a search for this compound in the database. So there's a particular chemical formula appearing at a particular address within the chemical space. And if we do a search on this, we see that the top hits are quinoa, beetroot, spinach, and I see all the botanists in here are nodding their heads because they're all part of the Amaranthaceae family. So apparently this compound is a compound that shows up in that particular botanical family. So this is the kind of exploration that we can do. The Z-score reports that I talked about earlier are also available for the dark matter side. That allows us to kind of target particular molecules of interest in order to do further exploration. So I believe that with that, I'm going to sit down.
Thank you. I want to thank Tracy, Steve, and Qiming. Unfortunately, we're out of time. I can't ask you any questions, but thank you all so much for this incredible work. Another round of applause for them, please. It's really cool. I wish we had more time to talk about it, but I know at lunch and during networking, you'll have more time to ask them questions. But it is so interesting